Okay. Primero quiero agradecer a Monseñor Marcelo Sánchez Orondo y a Eddie Robertis por organizar este meeting que apunta muy bueno. Bueno, vamos a ver la primera diapositiva. El título es How Glycans and Glycoproteins Provide the Cell with Information on the Folding Status of Protein Moieties. As you may know, about 40% of all proteins synthesized in mammalian cells follow the secretory pathway. About 80% of them, that is 30% of all proteins, are N-glycosylated in the lumen of the endoplasmic reticulum, and glycosylation is initiated by the transfer in most species of a glycan containing 2-N-acetylglucosamine, 9 mannose, and 3 glucose units from a polyprenol derivative to an asparagine residue in the protein. The constancy in the structure of the glycan transferred in most species is in deep construct with the huge variety of glycans that we find in the fully mature glycoproteins in the secreted proteins or in the plasma membrane. And this is because the glycans are processed by the removal of monosaccharides and addition of other monosaccharides all through the secretory pathway, mainly at the Golgi apparatus. Processing of the glycan starts in the lumen of the ER by removal of the 3 glucose units and some mannoses. But the lumen of the ER is the subcellular sites where glycoproteins adopt the final tertiary structure, and in many instances also the quaternary structures. And glycoproteins that fail to adopt the correct native structures are prevented from the exit to the Golgi, and this is called the quality control of a glycoprotein folding. And glycoproteins that never attain the native structure and translocated back to the cytosols where they are proteolytically degraded in the proteasome. This is called ERAD, for endoplasmic reticulum associated degradation. There are two issues here that have interested my group. And the first is, how do cells know if a glycoprotein is A, already correctly folded so as to allow its Golgi exits, or B, is it not yet correctly folded or has to be retained in the ER for further folding tries? And two, how do cells know that a glycoprotein is unable to reach its native structure and that it must be proteolytically degraded? As you see at the bottom, glycans have the onset. This is, the last one is not a trivial question, because folding intermediates are structurally indistinguishable from irreparably misfolded glycoproteins. So how can the cell know why to degrade one and retain the other for further folding tries? Well, glycans have the answer for that. And the first indication that the glycan is used by the cell as a barcode for the folding status of glycoproteins. And this is this is oh God. This is the oligosaccharide transfer. This is the blue ones are the 2-N-acetylglucosamines. Then you have the 9 mannose and 3 glucose residues. In the ER, the 3 glucose are removed, and also 2 mannose may be removed. And the first indication that the cell uses the glycan as a barcode for the folding status of glycoproteins came several years ago when we detected the presence of rather unusual glucosyl transfer transferase in the lumen of the endoplasmic reticulum, and this is the proteins of the properties of some of the of this enzyme. This is called the UGGT for UDP glucose glycoprotein glucosyl transferase that behaves as a sensor of protein conformation. The reaction catalyzed by this enzyme is once the 3-glucose have been removed by glucosidase in the lumen, this enzyme adds a single glucose to reconstitute the glucose 1 minus 9 N-acetylglucosamine protein. Well, it requires calcium for activity. It's highly specific for UDP glucose. It's quite large, about 1,500 amino acids. It's a soluble protein. But the most important protein is that the property is that the protein moiety has to be a non-native conformation for the activity, and I will show this property in very old and simple experiments. I don't know if you can see that. The reaction is the enzyme plus UDP C14 glucose and an acceptor. Here at the bottom, if you put the glycan link to a single amino acid, asparagine is not an acceptor, but if you put the same glycan link to 8 molar urea thyroglobulin, is a very good acceptor. And in the next slide, you will see, okay, if you use ribonuclease B, which is a glycoprotein, it's not an acceptor. However, if you put the same 8 molar urea denatured ribonuclease B, it's a good acceptor. Okay. Well, what are the structural features recognized by this enzyme? That's quite difficult question to ask, because folding intermediates are by definition unstable, and permanently misfold glycoproteins are a mess. So it's not easy to answer what are the structural features recognized by the enzyme. So what we did was to synthesize a peptide containing 64 amino acids, and we chemically added a glycan in cysteine in position 7, and we synthesized this peptide with different lengths. You see that the hydrophobic amino acids are in between amino acid 28 and 38, and the highest score for BIP is in the position 32. BIP is classical conventional chaperone of the ER of the HSP70 family, which recognizes heptapeptides with alternating hydrophobic and hydrophilic amino acids in an extended conformation of the protein. So fragments containing up to 40 amino acids had a random coil structure. That is, they had no structure at all. However, on increasing length, this neoglycoprotein adopted what's called a molten globule conformation. That is a conformation which has collapsed, loosely packed conformation, in which the amino acid side chains are not buried inside the molecule but facing the solvent. And we tested this neoglycoprotein as an acceptor, and you see, I don't know if you can see, the kinetic efficiency is almost nil until you reach a length of 40 amino acids. Then when the molecule reaches the molten globule conformation, it behaves as a very good acceptor. And then with increasing length and it approaches the native conformation with 64 amino acids, the kinetic efficiency decreases sharply. This on the left, on the right, you have the fluorescence of a reagent called ANS that is used by protein chemists to detect the exposure of hydrophobic patches. And as you see, the kinetic efficiency, the pattern of kinetic efficiency versus the chain length coincides exactly with the fluorescence of ANS. ANS is 8-anilino 1-naphthalene sulfonate acid. So this indicated that what the enzyme recognizes is the exposure of hydrophobic patches in the molten globule conformation. Molten globule conformation are adopted by proteins in the last stages of folding. So as you may recall, the highest score for BIP was in the position of about 30. And so we tested the ability of these protein fragments to increase the intrinsic ATPase capacity of BIP, yes. And you see the fragment that has 40 amino acids is more potent simulator of the ATPase activity than the molten globule fragment. This is because the sequence recognized by B has to be in an extended conformation. So this showed that first, if you can imagine that synthetic chaperones emerged into the ER lumen, first it would be recognized by BIP, and then as it collapses and adopts its molten globule conformation by the glucosyl transferase. And so this enzyme, 2 months ago, a paper appeared in PNAS by Italian researcher Pietro Roversi working in Oxford, where he solved the structure of this enzyme by X-ray diffraction and cryo-EM microscopy. And the enzyme appeared to have 7 different domains. There are 2 beta sandwich at the end of the arc, and they hold the catalytic domain. And then there are 4 thioredoxin-like domains in the middle of the arc. And one of these thioredoxin domains exposes an hydrophobic surface, and the authors speculate that domain is the one recognizing the substance. And what is remarkable about this enzyme is the extreme flexibility of the protein. And so the authors speculate that this flexibility allows the enzyme to recognize different protein substrates of different shapes and forms. This is illustrated by this in this. So the ER has 2 lectins in the ER resident proteins that are lectin. One is called calnexin and the other is calreticulin. Both are lectins that specifically recognize the glucose 1 -9 epitope created by this glucosyl transferase. And this is the structure of calnexin. It has an N-terminal domain, which is the lectin domain, and then has a long P domain rich in prolines that sort of embraces the protein moiety having this glucose 1 minus 9 epitope. And we use this property to demonstrate that in vivo, this enzyme uses the last folding stages of glycoprotein as a substrate. And for that, we use Trypanosoma cruzi. Trypanosoma cruzi has the distinction of not transferring a glucose 3 minus 9 glycan, as most eukaryotic cells do, but only minus 9 N-acetylglucosamine 2. They don't transfer glucosylated oligosaccharides. So the formation it's of monoglucosylated oligosaccharides in this case is fully dependent on the presence of, on the activity of the glucosyl transferase. So we pulse the T-crucized cells with S45 methionine, and then lyse the cells and precipitate them with anti-crucipain antibodies. Crucipain is a lysosomal protease that it's almost a recombinant protein because it's 10% of the soluble protein of the parasite, and it has 7 disulfide bridges. And you see, you have here when we ran the sample in non-reactive gel, we have the fully oxidized crucipine on the left, and some intermediates, crucipine with intermediate number of disulfide bridges. However, when you use on the right anti-calreticulin antibodies, we only pull down the fully oxidized crucipine. As you may recall, crucipine recognizes specifically monoglucosylated epitopes that are created by the glucosyl transferase. So that means that the enzyme, the glucosyl transferase, has glucosylated crucipine molecules in which all or nearly all the sulfide bridges have been already formed. So yes, this enzyme acts on the last folding intermediates. So this is what's called the ER quality control of glycoprotein foldings. Here you have on the left the peptide inside the lumen of the ER. The 3 glucose are the 3 red triangles. Glucosidase 1 removes the more external glucose. Then glucosidase 2 removes the second one and creates this glucose 1 minus 9 epitopes. Then this epitope is recognized by calnexin or calreticulin that behave as non-conventional chaperones because the interaction of the glycoproteins, the folding intermediates with calnexin or calreticulin increases the folding efficiency by preventing aggregation. And also, these lectins are associated with the PDI, a protein of ERP57, which is a PDI family member. And then the glucosidase 2 removes the last glucose, and so if the protein, glycoprotein is already folded, it goes to the Golgi. If not, it's reglucosylated by the glucosyl transferase, and the cycle, the calnexin calreticulin cycle, starts again. Glucosylation, deglucosylation until the protein is fully folded. This interaction, as I said, interaction with the calnexin and calreticulin increases the folding efficiency and prevents exit to the Golgi apparatus. For some years, we thought that the glucosidase 2, the glucosylation 2, which is the opposing activity to the glucosyltransferase, was a sort of naive enzyme that its only role was to remove the glucose unit. But then, when we started studying our work and the work of other groups, showed that it was not that naive. The glucosidase 2. You see here you have the oligosaccharide. It has the 9 monos, the 3 glucose. The glucosidase 2 removes the 2 internal glucose. Oh God. Okay. No. Okay. It's a dimer, it's a heterodimer, in which the alpha subunit has the catalytic activity, and the beta subunit has the retaining sequence at the C-terminus. But the most important thing of the beta subunit, is that at the C-terminus has here in yellow, what's called MRH domain. That stands for mannose recognition homolog domain. This domain is homologous to the domain that recognizes the mannose 6-phosphate epitopes in the lysosomal enzymes. It's present in the receptor that recognizes this epitope in lysosomal enzymes and drives the proteins from the Golgi to lysosomes. So we wanted to know if this, the presence of this MRH domain influenced the activity of glucosidase 2, and the answer is yes. This is just to show that we know all the genes that codify that code for this glucosyltransferase that form the lipid pyrophosphate glycan. So in yeast we can play with this genetically with the genes and make the cell transfer the saccharide that we want. So here we see after pulsing yeast cells with C14 glucose, and we see if, well, you cannot see because the lettering is quite, if we transfer glucose 2 minus 7, 6, or 5, after 15 minutes pulse with C14 glucose, we have increased amount of glycosylated glycans in proteins. This means that as the amount of mannose decrease in the glycans, the activity of glucosidase decreases also. If we only have this alpha subunit with the catalytic activity, we only have the same activity, not influenced by the mannose content. Then if we add the beta subunit in the plasmid, the wild type phenotype is recovered. And finally, if we express a beta subunit with a mutation in MRH domain, it's again we found this phenotype. So this shows that the MRH domain influences the activity of the glucosidase 2, and as the mannose content decreases, then the activity decreases also. So I will not go into this, but because short of time, but the number of mannose units in the glycan does not influence the activity of the glucosyltransferase. So if the mannose content decreases the activity of the glucosidase 2, but doesn't affect the activity of the glucosyltransferase, that would mean that upon decrease of the mannose contents, the half-life of the monoglucosylated species created by the glucosyltransferase would increase, and well, this is what is shown in in this experiment. So what we think that happens in the cell, first is that this is as I said, the glycan transferred from the lipid derivative. There's a very slow demannosylation and in the cell, very slow compared to the deglucosylation. I cannot pinpoint here the two mannoses that are removed but what we think that it's happening in the cells, and we start at the bottom, the 3 glucose are removed, then we have this shuttle between the monoglucosylated species and the deglucosylated glycans catalyzed by the opposing activity of the glucosyl transferase and glucosylase 2. And if the proteins falls quickly, it will go to the Golgi directly. However, if it has a difficult folding, then some mannose will be removed. The equilibrium will tilt to the left, and again we have the shuttle between the monoglucosylated and the glucosylated structures. And if it's even more difficult for folding processes going on, it will lose the second mannose residues, and we have tilting to the left again. So here, I couldn't show you, but the removal of the second mannose residue exposes alpha-1,6 mannose residue that is especially recognized by this a protein called OS9, which also have a MRH homologous domain, and it's part of the protein complex that drives the misfolded protein to the cytosols to be degraded by the proteasome. So, well, I will hurry up. This is part. The conclusion is that the occurrence of monoglucosylated glycans indicates that proteins are not yet properly folded, that they must be prevented from Golgi exit, and that their folding process must be assisted by the unconventional lectin chaperones calnexin and calreticulin. And B, that slow removal of mannose units provides a time clock that indicates that a difficult folding process is going on, that the glucosyl transferase must help the folding process with an increased interaction with the lectin chaperones, and that if native conformation finally failed to reach the misfolded, the misfolded glycoproteins must be proteolytically degraded. Thank you very much.